Hey guys, thanks for joining me in another IGCSE biology revision video. Today we're going to be going through some MCQ questions on enzymes that have come up in the past. So I want you to, what I'll do is I'll just flick through the questions. We've got 6 questions, and as I do so I want you to pause the video and read the question and come up with your own answer. Each question shouldn't take you any longer than 40 seconds roughly. So let's just go through each of the questions here. So here's the first one, and here's the second one, here's the third one, here's the fourth, and fifth, and sixth. Alright, so let's just go back to the first question and we'll have a look at the answers. So this question tests your knowledge about the relationship between enzyme activity and temperature. And the obvious answer here would have been A, because you can see that the graph increases in activity from around 0 to 36 degrees, and therefore as the temperature increases to around 25 degrees, or if not slightly more than that, the reaction definitely speeds up. Every other option did certainly not make sense. B, for example, suggests that from 10 to 20 degrees the enzyme stopped working, but we know that's not the case because there is certainly enzyme activity going on there. Between 35 degrees and 45 degrees, again, the enzyme is definitely working. It's only up till maybe around 50 degrees or more than that, the enzyme completely denatures and stops working, so that's not the correct answer. The optimum temperature for this enzyme would have been around, let's say around 36, and definitely not 40, and so the correct answer had to be A. So what statement about enzymes is correct here? And this really takes you back to your fundamental knowledge about enzymes. And so how it works obviously is that you've got the active site, which has a complementary shape to the substrate, and the idea that the enzyme's active site has a very specific shape that only fits one substrate, more or less, is the key to enzyme specificity, and you should be aware of that. So the answer, of course, would have been D here. The other options, again, did not make sense. Enzymes become part of the product. No, that certainly is not the case. The products leave the enzyme straight after. Low pH will always reduce reaction rate. We know that's not the case because some enzymes have a very low optimum pH, in which it's the pH that the enzymes work best. For example, if you take a look at some of the enzymes in the stomach, you should be aware that stomach has acidic conditions, and those enzymes have an optimum pH of 2 or slightly below that. And raising the temperature always increases reaction rate. We know that that is definitely wrong, because if at a certain point, if you increase the temperature too much, then that will break the bonds and the specific nature and the shape of the active site of the enzyme, and therefore denature it and causing it to become completely ineffective. And so question 3, why does excessive heat reduce enzyme activity? You actually just covered that, and that's because the active site basically becomes destroyed or altered due to the excessive heat, because the specific shape of the active site of an enzyme is really dependent on the molecules and how they're bonded together, and if you're increasing the temperature to a very high degrees, then those bonds become broken and ultimately alter the shape of the active site, and that specific shape is really important for the enzyme to work. So here the answer would have been A, which is really just testing you on the fundamentals of your knowledge of how denaturation happens. It would have been easy to confuse the answer with B. It alters the shape of the reactants in the products, but certainly that is not the case. The reactants in the products remain unchanged. It's the active site shape that is altered from the high temperatures in denaturation. Okay. So next question, 4 test tubes were set up in the table as shown. Okay, so in which test tube would starch be broken down the fastest? And here this comes down to your knowledge of the fact that every enzyme has an optimum temperature, and amylase, you find that in the human body, and generally in the human body, enzymes will have an optimum temperature around 35 degrees. 5 degrees would be way too low, so automatically it would point us into the direction of A or B or D, having the optimum temperature of around 35 degrees. Now, we know that starch is digested or catalyzed by amylase, and you should know that from your other topics. But so we definitely need the presence of amylase in order for this to happen. So the answer would have been B. Now the answer is not D because D includes boiled amylase, and boiled suggests that you are putting amylase through very, very high temperatures, way above the optimum temperature of around 35 degrees, at 100 degrees or below, above that obviously, and that will denature the amylase. And even if you had amylase, it's denatured, therefore it wouldn't actually affect or break down the starch, and you therefore need fresh amylase that's actually working and not denatured, and that would point us towards the direction of answer B in that particular question. Right, so here this graph again is about the relationship between temperature and the rate of reaction of enzymes, in specifics of how the molecules behave when the temperatures go up. So, you know, we understand that this graph shows that the temperature goes up from around 20 to 30 degrees here and starts falling down above the apex and eventually denatures. So what causes the rate of reaction to change or increase from 20 degrees to 30 degrees? Okay, so for any reaction to happen, particles need energy when they collide. So there's two key things here when you're talking about reaction rate. One, how much energy do those individual particles have when they are colliding together, and two, how often are they colliding to begin with? And so what happens when you increase temperature is that individual molecules, whether it be the enzymes or the substrates or the products, they just have an increased amount of energy, and when that happens they move around a lot quicker. What that means is if the molecules themselves are moving around a lot quicker, then the rates of them colliding together will increase, and also when they do collide, most of those molecules have enough energy for the reaction to happen because of the increase in temperature and the individual molecules having more energy. And so therefore it is a combination of an increase in kinetic energy of the individual particles and the increase in frequency of collision between those particles. So here the answer would have been A. So this final question is a question that is sort of testing knowledge on the relationship between the pH and enzyme activity. And so this is a very simple question where they just get you to understand what goes where. The y-axis, just like the temperature graph that we looked at before, would be the rate of reaction, and the x-axis in this case would be the pH. And you can notice that the optimum pH is at this apex here. When the pH goes far above or below that, then enzymes end up being denatured. That's why you have the graph going to its minimum or to 0 there. So the answer again would have been the y-axis being the rate of reaction and the x-axis being the pH. So again the answer here is A. So I hope this video helped you guys, and if you liked the video, please like, share and subscribe. Otherwise, please check out my Patreon channel where I've got a whole heap of MCQ questions that are organized into topics just like this, and a lot more for enzymes as well. So it's really going to be a helpful tool for your revision, and otherwise I will see you in the next video.